record to the cloud. Thank you so much, everybody, for joining us today. My name is Tommy Soares. I'm Assistant Director of the Purdue Institute of Inflammation Program. I am delighted to welcome Dr. Tim Richardson from the Indiana Bioscience Research Institute. And Tim, how long have you been with the IBRI?
I've been at the IBRI a little over a year now.
Okay, a little over a year. And IBRI is located, as we were just talking, downtown 16 Tech in Indianapolis, Indiana, right? And so tell us, Tim, tell us a little bit about your background. Are you originally from Indiana?
No, I don't know how far you want to go back. So my accent probably doesn't reveal it, but I was born and raised in Little Rock, Arkansas.
Okay.
I will date myself a little bit. Bill Clinton was governor when I was in high school. Yeah.
That's great.
Long story, but I ended up at the University of Minnesota. So I have experienced long, hot summers and long, dark, very cold winters.
Yes.
One of my favorite things about the chemistry department, University of Minnesota, fourth floor is where all the organic chemistry was. When the temperature would get about to minus 20 outside, I would open up the GC ovens to try to provide a little more heat to the lab. Yeah.
That's too funny because I remember during my PhD here, I would look forward to, I had to do a lot of protein work and I did everything in the cold room. And on a hot summer Indiana day when it was all so humid and hot outside, I'd look forward to spending the day in the fridge. That is awesome. So it got really cold, eh?
Oh yeah, I had one of those minus 100 thermometers hanging right outside the window.
Oh God. Oh geez. They get long, those winters, don't they? They really get to you. So, I mean, other than the better weather, maybe, what attracted you to come to Indiana and seek out this position?
Well, I spent most of my life actually in the central time zone. I think I actually am about to cross that threshold now that
Okay.
Indiana is on the Eastern time zone.
Yes.
So I can say I'm sort of the middle of the country at heart.
Okay.
Yeah, I did actually do both my grad work and my undergrad at the University of Minnesota. I was actually in the biochemistry department for my undergrad. This will
Okay.
date me a little bit as well. I did undergraduate research basically doing thermal calorimetry with proteins and how small molecules, basically measuring the thermodynamics to measure delta G and Ki of molecules binding proteins.
Yes. K on, K off
Yeah.
rates.
And we actually built those calorimetries by hand, like with Radio Shack parts, basically.
Oh, wow. Wow. Cool. Cool. So you really know what you're doing in that area.
Well, I've always had kind of a love-hate relationship with biology. I love learning about biology, but biology is super frustrating because you never know what you have when you're doing biology, right? And I distinctly remember one day looking at a flask where I had made this molecule, nice white powder. I could take an NMR, I could take an LC, I could take a mass spec, and I knew what this thing was. So that was really the decision point.
Yeah.
Moving into being able to make new molecules. And I've always been the kind of kid that played with Legos, tinker toys, right? So when I found out that you could build molecules, you could play with Legos with molecules, and there
Yes.
might be some sort of chance that you could help someone by discovering a drug, that's when I was hooked.
Awesome.
From that point, that's why I joined the Rognovsky lab at the University of Minnesota. Did macrolide antibiotic synthesis there. So if you're not familiar with those structures, it's polyol, polyene, macrocycle. Learned a lot of organic chemistry synthesis, being able to put those molecules together. A lot of strategic thinking goes into natural product synthesis. And then I did a postdoc with Dave Evans at Harvard. And there I worked on another macrolide antibiotic, but a peptide. So it was vancomycin. It was about a decades long project in the Evans lab. I was on the tail end of it, worked with a very talented graduate student, Wes Trotter, and he and I finished up that synthesis. And then transitioned to Eli Lilly here in Indianapolis. I believe I applied to every possible pharma position in the Midwest. And I think I picked the only company where I was able to spend 20 years of my career. If I had gone anywhere else in the Midwest at the time, like Park Davis, I mean, Upjohn, the list goes on and on. Basically, none of those groups are here in the Midwest anymore. They've either gone to the East Coast or the West Coast.
And so that's really interesting. So that the stability was really in the company itself, not necessarily because you would have moved away. Would you have preferred to stay in the Midwest than in and around the area?
Yeah, I mean, for family reasons. I mean,
Sure.
like it or not, I mean, Indianapolis is a great place to, establish yourself and raise a family.
Yeah, that's awesome. That's really great to hear. And I'm so happy to hear that perspective because we don't hear it all the time, Tim. And to tell you the truth, I love visiting Indianapolis, but I'm a visitor there. I don't live there. And so to live and work there, it must be a different culture. And it's good to hear you say that. So tell me a little bit about what attracted you to then go join forces as the scientific director of molecular innovation at the Indiana Biosciences Research Institute.
Well, in 20, right before the pandemic, actually, it was Alan Palkowitz and Bruce Lamb received this NIH funding. So the National Institute for Aging funded a drug discovery center at the IU School of Medicine. So there are a number of PIs that have their primary appointment in the School of Medicine and a secondary appointment at the IBRI. So my primary is actually in clinical pharmacology at the School of Medicine. And my primary responsibilities are with this Treat AD Center. So it's an academic drug discovery center focused on the neuroinflammatory components of Alzheimer's disease. We have a good collaboration with another NIH-funded center called the Model AD Center. As the name suggests, it is a center that is focused on building mouse models where these mice are genetically engineered to have some of the same risk or protective genetic variants that are found in human disease. So the Treat AD Center and the Model AD Center work together. Treat AD Center focused on small molecule drug discovery, and then the Model AD Center on building these animal models where we test the molecules in those models for pharmacokinetics, pharmacodynamics, efficacy, and models that we believe are more translatable to human clinical trials.
So neat, and such an interesting look into the drug discovery world, where you do have a model arm and where you can test the chemistries that you're building on the side, right? And so I guess and we could go, like, we could peer a lot deeper into that. Tell me a little bit about, on the molecule side of things, how things have changed since you finished your PhD to where are we now, especially with computational methods, cheminformatics, and machine learning techniques for modeling and fitting, molecule fitting programs, algorithms that have been coming about. Has that been an incredible change in the field, or has it been modest because we are yet perfecting these computational models?
I'm a computational fan. So my joke, I guess, is electrons are less expensive than molecules. So you can always take a computer, plug it into the wall. Um,
Yes.
some people, are a little bit standoffish with computational uh, strategies. I think because, they're, so if you ask me, how good does a model have to be for me to be able to use it? And my answer to that, well, it has to be just slightly better than my brain, right? Because I mean, if you're sitting down with a piece of paper and the old days, you're just printing stuff off with ChemDraw and holding up to a light and seeing what overlays, right? Um, you're still using a computational model. It's just what's in your head, right?
Sure.
So I'm a big fan of using computational strategies. And the way I like to think about it is, your hypothesis building. You're not testing hypotheses with computational strategies, but you're using those tools to define hypotheses. And so I do believe that we are at a bit of a tipping point with uh, artificial intelligence, or really machine learning type strategies. I don't think they're as new as people like to make them seem. So, we've been using, random forest or, SVM models for, more than a decade now. I do think they've gotten better as computational power has increased. One of the things that I did have to do when I transitioned out of Lilly into academia is I didn't have access to the, in-house systems that I had access to at Lilly. I didn't have access to the Lilly database. I didn't have access to a system we called Mobius, which is basically in-house data handling capability. So I might do a little bit of advertising here. Uh, to put it at a high level, we needed a place for our data. So we needed a database. I evaluated a number of different options out there. We ended up going with CDD Vault, which is collaborative drug discovery. It's a cloud-based data system that gives you access to all your data, in vitro assays, in vivo assays. And then there's a little bit of data analysis, visualization capabilities to CDD as well. But what we decided to do is layer on top of that a computational and informatics infrastructure that Schrodinger makes available called Live Design. And that gives you the ability to analyze data, visualize data, and put data together in table formats and make correlations between data sets. So you can understand the directionality of your structure activity relationship studies. Another nice thing about Schrodinger is that also gives us access to their drug discovery platform. So Maestro, Glide, and there's a dozen or so capabilities that come along that allow you to model how compounds interact with proteins.
And I think what's interesting about the way that you're pointing it out, is that there are certain advantages that you had in industry that you might not have now in your role in the IBRI. Maybe it's good at this point to tell the people that are online, that are listening in, what is this IBRI? Is it an industry partner? Is it academia? What is it? And what should we consider it?
Yes, it's a not-for-profit discovery center. Its nucleus is the diabetes group there. and they've had a long-standing relationship with Lilly. One of the things that the IBRI is known for is their iPS-derived beta cell platform. That platform is being expanded out to additional cell types as well. One of the goals of the IBRI is to facilitate collaborations between, the academic groups, Purdue, Notre Dame, IU, and then help establish biotechs, and then establish relationships with pharma, not just Lilly, but AbbVie, other pharma as well. I think especially in a post-COVID world where we're all, connected on Zoom and et cetera, being able to collaborate seamlessly between various groups is much easier.
Yeah.
One more thing I will add to that is, within the Treat AD Center, within the School of Medicine, we have just enough capacity or budget, to be clear about it, to pay for things like CD Vault and Schrodinger and, all the, what I would call, bread and butter infrastructure that you need to do drug discovery, but just barely. So one of the goals of the IBRI is to put these capabilities in place and then make them more generally available to PIs at IU, Purdue, and Notre Dame, so they can access things. And it can be things you may not appreciate just how important like sample management is, right? So, you make these compounds and you can have, in a typical, SAR, structure activity relationship study, you'll easily generate a few hundred compounds, maybe even 1,000 compounds. Big bad projects at Lilly might generate 10,000 compounds.
Right.
So you can imagine trying to handle, even a few hundred compounds on your bench. And of course, you can't just, like put that in an envelope and mail it to your biologist, right? And your biologist would prefer it as a DMSO stock solution that they can just make,
Yes.
right away, right?
Right. Right.
Or, if it's in vivo biologist, you need to provide it in a vial in a certain state that they can put in a formulation, dose it in an animal. None of that is easy.
Right.
And so you really need to put infrastructure in place to do that in a way where you have all the quality control. So that's, there are these sort of bread and butter aspects of drug discovery that don't look innovative, but are very clearly needed to be innovative.
Right.
So that's a major goal of the IBR right now, is to put those bread and butter drug discovery capabilities in place, not just for small molecules, but large molecule as well. Another thing that the IBRI has access to is a phage display platform to select for antibodies. So not just small molecules, but antibodies as well.
Cool. Phage display being such an awesome technique, right, for drug discovery in terms of identifying binding partners and being able to move the piece forward that way, the line forward. Tim, what do you think is the most or how do you define success in your center there? I mean, what drug discovery has so many moving parts, so many moving pieces, and is it really about the what scaffolds you guys come up with, or is it really moving it down the translational line to de-risk it enough to get it through FDA and commercializing?
Yeah, so define success. Well, let's start with, I'm going to answer it I'm going to try to answer a number of different ways. I'm going to use the phrase stage appropriate success.
Okay.
So what stage are you in the drug discovery process or journey? It's probably a better way to say it. And this gets to questions like, to put it in more technical terms, like we could ask, what's a valid target? Or we could ask, what is a druggable target, right? Because ultimately, in order to be successful, you have to identify a target that's biologically valid and druggable through some sort of modality. Now, one way we could answer that is, well, a valid druggable target is one that's demonstrated its ability to recoup the cost of discovery, development, and marketing, provides value to the patient and the physician. Okay. Of course,
Yes.
by the time you get to that point, someone else has already beat you there, right? So by the time you define success at that level, right?
Yeah.
You're a me too, or you're so far behind that you might as well not even start the project. Okay. Okay.
Okay. Very optimistic. Yes.
Yeah. So let's work backwards from there.
Okay.
And I do think it's important to work backwards, because I can't tell you, like, the best projects I worked on at Lilly were ones where we actually sat down with physicians and talked to them about their patients. And we basically asked two questions. How does the patient present, and how do you treat them? And then where do you get stuck in that diagnosis and treatment process?
Right.
Because ultimately, the patient doesn't care how the molecule works, right? And actually, the doctor doesn't really either,
That's right. I was
right?
thinking that.
But
Yes.
you are giving the physician a tool to treat their patient. What the patient cares about is just getting back to their normal life, so their
Yeah.
life isn't disrupted by their disease.
Right.
Um, so if we take a step back from, is the molecule valuable, right? What does it do for the patient and the physician? And then, once we answer that question, and from the patient perspective, it's usually efficacy. So it's got to have some sort of efficacy, and
Yes.
it's got to have enough efficacy that it beats standard of care. It's got to be better than what's already out there. So it's got to have a significant amount of efficacy, and it's got to have enough safety so
Yeah.
that it also beats standard of care,
Sure.
right? From the patient perspective. And then from the physician perspective, um, does it give them that tool that they need? So when they run into those roadblocks with standard of care, is it another tool that can pull off, their shelf that provides them the ability to basically make their patients happy? Right?
Yeah. Yeah.
And then let's step back from there. I won't go through the whole development process, but let's go all the way to phase one. Okay. So phase one, it's a small study where you need to be able to demonstrate that you can engage the target safely. Usually you do that in a healthy volunteer trial. And actually, I'll point out here, the FDA cares a lot more about safety than anything else, especially in the early part of,
Right.
of, of development. Of course, you don't want to waste money. I mean, the FDA will let you waste money on bad hypotheses as long as you're, safe with the patient.
Right.
Okay.
Right.
So if, at phase one, it would be, have I designed a molecule that gives a certain exposure over a certain definition of target engagement. Okay. And so that's, enough concentration in plasma or the target tissue that you're actually engaging the biological and target that you intended.
Right. Right.
And then
And
go back from there. Do you want me to go all the way to the beginning, or?
well, you don't have to. I mean, I think I understand, or from what I gathered from what you're saying is that success is not necessarily defined as a trajectory, because there's so many steps along the way that could be seen as milestones for success, and that reaching those milestones and progressing through those milestones, you're making success happen on a constant basis if you can. And the molecules that don't make it through the milestones, you don't consider them successes, and they're put aside, right? They're taken aside out of the line. What do you say though, Tim, makes for a successful scientist in industry? What type of character, what type of persistence, what type of energy does this person need to have to succeed?
Well, let me answer that. I'll answer that at a philosophical level, and then I'll, you know, take a few steps down and just give you a few practical examples. So at a philosophical level, and I think this applies to really anything, not just being a scientist, is, uh, within any organization where you're operating, collaborating with other people, you need to be a valuable part of that organization. In order to be valuable, you have to create value, right? So the most successful people in any organization are ones who create value for themselves and for other people. Now you might think, well, I could just be completely utilitarian and create value for myself. There's certainly plenty of people who do that. I don't think they're as successful or as well liked. And you could, create value for yourself and actually harm other people. That's what criminals do, right? I mean, they go and they steal something from someone's house. So they created value for themselves and actually harmed someone else. And you can be
Right.
successful doing that.
Yeah.
But the most successful people are people who create value for themselves and other people.
Yeah.
You could also flip it around and say, well, I just want to be altruistic and create value for others. That's another path. I don't think you're as successful down that path just because you're going to get burned out. You're going to plateau. You need to take care of yourself at the same time you're taking care of other people.
Absolutely.
Another thing I'd point out about that kind of a framework is one thing I do think is challenging is, as you're creating value, if you have this eye towards creating value for others, you do have to be willing to give away things that you've created. And that can be hard sometimes. So I think that's the break point between someone who just creates value for themselves and someone who creates value for themselves and others.
So interesting that you say that. So for example, if I make a molecule and I pass it down the line to somebody else, is that kind of like, okay, it took me months to make this, now I need to relinquish this to you, and you're going to do whatever you say you are going to do with it, and I'm okay with that because it's helping you make progress in your area, in your field.
Yeah, to give you some practical examples. So as a medicinal chemist, when I first started at Lilly, just a single chemist and, my own lab, working on structure activity relationship studies, I created value by identifying scaffolds and then working out the SAR. And then I had to be comfortable with other labs or other chemists, basically jumping on that bandwagon,
Right.
right? And not being upset that this guy's making a molecule that I could have made, right?
Right.
Um, so,
Right.
as a single chemist, that would be the way to think about it. When I got my first associate, then it was making sure that that associate chemist had scaffolds they could work on. So I would concentrate on, the front running chemistry, the novel scaffold. And when I found, kind of what worked out, then I'd pass it off to my associate and then they would take off from there. When I got to the point where I was leading a chemistry team or a scientific team, I was working with the other scientists, the other chemists, making sure everyone had a place to work, within the problem sets of the project. And then when I got to the point where I was leading teams of teams as a group leader, it was working directly with the group leader and then making sure, obviously, the projects that they were leading were good projects and not crappy projects, right?
Right. Right.
And then doing, there's a lot of stuff you have to do to, just like in academia, you got to write grant applications. In industry, you got to convince the people who are paying the bills that yes, this is a good project, right? So I took care of a lot of that work that needed to be done with, therapeutic area vice presidents, functional group vice presidents, that yes, this team leader is leading a project that's valuable for these reasons, right?
Right.
So at
So
that point in
is
my career, it was mainly fighting for others.
okay. And is it still proposal based? Like, do you still have to write it, or do you pitch it to them in a presentation? How does that work, Tim, when you're at that level?
Every company is a little different. I would say in general, every company has some form of what I would just describe as portfolio management. Um,
Hmm.
so there's a whole portfolio of projects. They're at various stages. There's a budget, and they're trying to figure out, which are the best projects that are gonna likely transition to clinical trials.
Hmm.
And then, there are functional areas. So there's the ADME scientists, there's toxicology, there's chemistry, there's biology, there's the various therapeutic areas that all have a certain amount that they contribute to those projects. So a big piece of that portfolio management is actually decision sciences. So they actually have a group of scientists, and their whole job is basically helping management make decisions
Wow.
on which projects move forward and which ones don't.
How interesting.
Yeah,
So that's in a big, big company like Lilly, right?
right,
A smaller company, there is no such team. You are the team, right?
right.
Yeah,
And so
that's awesome.
a small biotech, the best small biotechs are ones who have a platform, either a technology-based platform or a biology-based platform. And they have minimum, 2 to 3 assets across a portfolio that
Mhm.
they're, bringing forward proximal to IND filing for a phase 1 trial.
Yeah. And so, Tim, with it seems to me that in your history, in your background, you had a really good view of academia, and you've had a very deep view now of industry, as you say, for the past 20 plus years, right? That you've been in industry. What do you wish you knew as you were leaving academia and going into industry? What is it that you would say to people who are thinking about going to industry who are now in academia?
I'll just, repeat some of the good advice that I got when I started. Uh, as much as it took me a long time to actually really absorb this, uh, it is a marathon, not a sprint. In fact, I think I would say it's more like a very long, varied hiking trip, like hiking in the Adirondacks, than anything close to a sprint. So any particular problem that you're on, and especially any sort of setback, there is no setback that isn't recoverable. So like if you're hiking along and you trip and fall or something along those lines, you can get back up, you can find the trail, and you can get going again. Um, One thing I will see, there is this sense that my job is going to be, quote, more stable if I go into industry. Um, I think that is true in terms of, perhaps you don't have to move, right? Like, don't have to go from one city to the next. But even if you go to Boston, right? I mean, there's 100 biotechs you can move to, right, without having to change where you live. Even within the size of a large pharma company, there's still changes in strategy, still changes in leadership, therapeutic area concentration changes, uh, even modalities. So small molecule, antibody, siRNA, peptides, even the focus around modality can change.
Yeah.
So you may go in there thinking I'm going to be a small molecule chemist and I'm only going to work in this therapeutic area. I guarantee you that's not going to happen, right? So I had to change a therapeutic area and even change modality a couple of times while I was at Eli Lilly. So you will run into these, career changing pivots that you'll have to do, even if you have to do that within a company.
So staying flexible is always a good thing, staying nimble. But I'm sure that's also, in a way, a challenge that you kind of, say, oh, here's something new that helped me grow. It's not like you stop doing the other stuff or you stop knowing the other techniques, but now you pick up new techniques on how to work with different modalities. And so I would think that that growth is good from a personal development perspective too, right?
Yeah, as a chemist, there's always a design piece. And that design, whether it's a peptide, a oligonucleotide, even an antibody, those design principles, the pieces and parts change, but those design principles and structure activity relationships, that
Yeah,
stays the same. It's modality agnostic and it's therapeutic area agnostic as well.
yeah. Is there anything you miss about working in academia that you don't now that you don't have access to or you don't have now?
I do think it's true in academia, you are much more the captain of your own ship. So, you have your own lab, you have to deal with your own budgets, you do have to publish, you do have to get grant applications. The flip side of that is you're much more in control of your own destiny. When you're in industry, you do have access to a lot more infrastructure,
Yeah.
and you don't have to worry about budgets as much. But you're definitely not the captain of your own ship. You do have to fall in line with what the goals and particularly the portfolio goals of that company.
Yeah.
Actually, it doesn't matter if it's drug discovery or you're making paint or sticky notes at 3 a.m. I mean, there's a product portfolio there, and as a scientist, you have to figure out how to add value to that product portfolio.
Yeah. Tell me something though, Tim. You're in this kind of hybrid institute now. You're living the best of both worlds as the way that I see it. Is that right? Is that a good perspective, or is it different now, kind of in this position that you have? Do you like it more? It sounds like maybe you have more control of the fate of your program.
Yeah, at this point in my career, it has definitely been a beneficial career change. Um, one of the things I do really appreciate about this particular position now is just when you're in another aspect of being in industry is everything is really trade secret that you work on, right? Until there are patents that are filed and then publications much later. But now as an academic, I can literally call people up on the phone and they'll actually talk to me, and I can just tell people what I'm working on. Right?
Yeah.
I don't have those sort of confidentiality constraints
Yes.
that I used to have in big pharma. So having a much wider canvas to paint on, that has been very freeing, and that types of science, types of targets, types of biology.
That's interesting because I'm sure that people on the phone or people on this call have wondered about that. Is it restrictive? How restrictive? How much freedom? And that you went through the traditional academic route to get your PhD and so on and so forth, and all your experience, but a lot of your experience afterwards in industry and coming back now to this academic like position to work on this NIH project for Alzheimer's. And I'm just wondering if that is that constriction the case, or is it that in industry they do care about allowing certain freedom for your scientific input and scientific thought to be brought to the table, and that's one way to succeed in industry is by making good suggestions and how to move forward.
Yeah, the overriding concern in any industry, I think, but in pharma in particular, is how is your science contributing to the portfolio? How is it moving projects forward?
Right.
So in, like a small molecule drug discovery time frame, getting back to that stage appropriate, if it's early stage, you have a idea, a target, you have a hypothesis around that target, you're going to have to engage it with a molecule. So
Mhm.
the very first stage is ligand identification, some sort of screening process, some sort of way that you find that starting point for chemical design. And then, at a middle stage, how are you designing that molecule so it actually has exposure in an animal model? Because you have to test that hypothesis preclinically first. And there's no way to do that outside of whole animal physiology. As much as we like to think we could do everything in vitro. Um, and then once you do that, of course, if you're going to get it into humans, you have to figure out if it's safe.
Right.
And then you get into the tox realm. And my joke at that level is toxicology is whatever your toxicologist says it is. Right. So, it can be a little bit challenging in the high dose tox world to figure out what is causing the tox.
Sure.
But there you're basically looking for a molecule that engages the target at a level where it's safe enough that you can dose it in a human clinical trial
Right.
and give your clinical pharmacologist a comfort level and that those patients are safe when you give them the molecule.
So interesting, full circle back to your biophysical analysis, your binding affinities, K on, K off, equilibrium rates, and how interesting that, full circle, because you need to have those basic techniques, or at least understanding how that relates to the eventual dosing and how the affinity and stoichiometry of binding at that molecular level really will end up defining how much concentration you need to put to dose that target, right? And that patient. And it's interesting how a lot of these fundamental pieces don't change, and many of us, I think, even still take for granted that I leave my Tylenol or Advil or whatever bottle in the medicine cabinet for months, years, Tim, and this thing still works. How is that? That's like a miracle of life. And so to think about how many chemists does it take to really translate a molecule into being able to have a pill that sits in the cabinet for years there and still works? How that whole process requires such a village of people to bring it through. You've given us a good glimpse of some of that. You're so knowledgeable about the processes, especially here at this preclinical stages of development, right? Is that is that um, is that something that in industry versus academia, you see more legs, meaning that molecules that you do bring about, and I know now at the center that you're in, the purpose is really to bring chemistries up through to the clinic, right? And being able to start testing them out after they've been vetted through these models that you guys have in your center. or the other modeling center, I should say.
Yeah, it is one of the daunting things about drug discovery, but one of the that appeals to me uh, is the breadth of science that it does touch on. So, I mean, as you implied, everything from solid state physics, basically, uh,
Yes,
all the way to basically human sociology. I mean, at some point, you have to have a patient that's willing to actually make this drug a part of their life, right?
yes,
So it covers that whole gamut. I think probably the best way to answer your question, perhaps another little bit of advice for anyone starting their career. If you're going to be a chemist in drug discovery, you have to interact with a lot of different types of scientists, a lot of different types of sciences. Um,
yes,
so always remember that each individual that you interact with, you're probably going to be collaborating with them, at some point in your career on some sort of problem. So another piece of advice I would give is be kind to everyone that you work with, because you're probably going to be working with them on many different problems throughout your career.
no doubt, no
I
doubt.
always try to, since I am I'm a trained chemist, so I always try to speak from the perspective of the molecule and how the molecule engages the target, right? And so from that perspective, if my formulation scientist is saying, this thing is brick dust, it's got to be more soluble, right? Or
Yeah,
if my ADME scientist is saying, this is a wicked CYP inhibitor, you have to fix this problem,
uh-huh.
right? Or on down the line, I could come up with 100 different problems. You have to know enough about their science to understand what they're telling you and to
Yes,
be able to ask the right questions, right? And to be able to trust them and work well with them, right? So, I mean, that's another piece of advice I would give to people is being able to understand the types of scientists and the sciences to that point where you understand what they're saying and you can ask the right questions.
so true, so important, anywhere and everywhere, and that's a great piece of advice for life. Because I think there's so much that we do end up doing as a team, right, and relying on other people. Um, but understanding the language is a big barrier, right? Is being able to communicate clearly so that they get just enough information that they need so that they can move forward and not give them all, because they are not the chemists that you are, or they're not the scientists that you are, and they might not get it all right. It takes finesse. It takes some experience there. Uh, Tim, it's so good to talk to you, and I want to open it up to see if anybody in the call has any questions. If you do have a question, we ask that you either unmute yourself or you can type your question in the chat. Um, Christina Ferreira is asking, can we ask questions? Yes, please go ahead, Christina.
Hi, are you hearing me?
Yes,
Yeah,
perfect.
great. Nice to talk to you, Tim. Thank you, Tommy, for the opportunity. Yeah, I was very curious to get your opinion about the role of mass spec in synthetic chemistry. I think you may be aware of this high throughput reaction screening system using DESI at Purdue. So I would be very curious to know, your opinion, how useful this system is, you think, for like preclinical studies of synthesis.
And so I think the biggest advantage of mass spec technologies would be in the what I termed ligand identification, so screening techniques. One of the biggest advantages of mass spec is you can set up what I would describe as label-free assays. So you can, there are a lot of different assay strategies where you have some sort of reporter substrate or some sort of reporter assay, and it's really not biologically relevant. It can be very biologically irrelevant, right? So the advantage of mass spec is, you're able to measure the substrate directly. And also you get a mass, right? So being able to measure the mass is really good too, because you have at least have some data to support that what you measured is actually what you measured, because you have additional data to support that. You can also flip it around. So DESI allows you to in assay wells or even at a tissue very quickly sample, right? So that's one of the advantages of DESI. You can actually flip mass spec around in a assay context and assay for the small molecules that engage the target. It's known as a affinity selection mass spectroscopy. So there you're incubating your target protein with the molecule or mixtures of molecules. And then you're using mass spec to basically figure out what molecules actually stuck to the protein. But again, it has the same advantages. It's label free, so you didn't do anything goofy to your protein. You didn't do anything goofy to your molecules. You're just simply measuring what molecules stuck to the protein. And you get a mass, so you get extra data that proves the molecule you think bound the protein actually did bind the protein.
Great. I have one small question. I don't want to,
Go
use
ahead,
up all the
Christina.
time, but I would like to know your opinion also about this simulation chemistry. When you use like quantum calculations or you simulate molecule structures or reactivity, if this has a future, what's your opinion about what is being made today? Something like this.
Like I said, I do think we're at a tipping point. Now I'm a little far along in my career, so I'm not sure I can take advantage of that tipping point. But I actually do have some collaborations where we're trying to use machine learning or AI type strategies. It's mostly around predicting activities, but the same neural network type, graph neural network type strategies can be used to map out reaction pathways and even design synthesis routes and predict whether reactions will be successful or not. Of course, one of the major limitations are these models do have to be trained by data. So one of the biggest, I think, hurdles is having good, solid data sets to train these models, right? And you might think, well, you like at a company like Eli Lilly, you've had electronic notebooks for over a decade. Don't you have enough data? But you can imagine, I mean, when I was at Harvard, we would call it the Boston Harbor workup when the reaction didn't work. You can imagine in the notebooks, when a reaction doesn't work, I mean, just
Nobody published, so you don't have
there's no data, right?
negative
Yeah.
database, which is important.
Training the models, you have to have a kind of a balanced data set. You have to have some positive results and negative results. So if you have an imbalanced data set with not enough negative or not enough positive, it's basically not a good enough data set to train the models.
Yeah, well, that's very good to know, your view of these topics. Thank you so much for the opportunity.
Yeah, thank you. And thank you, Christina, for asking questions. Anybody else that would like to ask a question? The microphone is open to you. And, it's such an interesting perspective here with machine learning and what computational biology, computational chemistry, cheminformatics, all of these fields, as you say, are, yes, they're going to help us with reaction and planning, and even I think in toxicology, and even in looking at other drug discovery modalities that are data intensive, and we're not necessarily always best suited to look and find patterns. I think, you know, the other thing, Tim, is we haven't spoken about really is, you know, what happens down in clinical trials further down the line, because there's a lot of inefficiencies that do happen down the line too, that could kill a molecule, because there are, you know, the data is not adequate. Um, and so I'm thinking, you know, like, could we do some engineering in there to improve the efficiency? Yeah, Six Sigma type of black belt to improve the efficiency of clinical trials or even preclinical trials and what we can do. But I think we have it down packed. Industry has done a really good job at giving us a good formula for the preclinical tox ADME work, right? I mean, you guys have that. You know exactly what needs to be done to move a molecule down the line, right?
Yeah, the ADME talks, one of the, that in the course of my career, the last 20 years, the ADME tox sciences have made leaps and bounds. I would say we're at the point where there's really no excuse for a molecule to not have exposure in your phase 1 clinical trial. If you fail, you didn't do your ADME tox very well.
Right, right. And I think going back to, you know, Christina's point too, is the use of mass spectrometry more and more. If we can figure out how to get a little bit more or more understanding of the tool that we have at our hands. I mean, doing screening with mass spec is incredible, because not only do you know which molecules bind, but you also have some idea of stoichiometry all in one measurement, right? And it's really something that could be really, really valuable if more and more people continue to. Are you guys there? Are you hearing me?
Yeah, Tommy, you're breaking up a little bit.
Firing off here. I'm looking at the hour. We're at the top of the hour, and I do thank you so much for your time, Tim. I thank everybody on the call today too who was able to join us. Really, what a insightful look into the world of drug discovery, into the world of what you're doing now in Alzheimer's and your experience, Tim. We thank you so much for everything that you've given us today with your advice and your guidance as well. So thank you.
All right, wonderful. I had a good time.
Yes, and thank you everybody else, and hope to see you at the next one. We'll catch you soon. Stay tuned.
Bye.
Bye-bye.